Good afternoon to our respected Director General, dignitaries, delegates from Argentina, esteemed faculty members, researchers, and dear students. Welcome to day 2 of International Academic Lecture Series organized by Amity Food and Agriculture Foundation at Amity University, Uttar Pradesh. Yesterday, we explored innovation in science education and interdisciplinary perspective that strengthened global academic collaboration between India and Argentina. Today, we continue this enriching journey with a lecture that highlights the transformative power of molecular science in addressing urgent public health challenges. With great honor, I would now like to invite Dr. Nutan Kaushik Ma'am, our Director General AFAF, to please welcome the guest for today's lecture.
A very good afternoon to all of you. On behalf of our Honorable Founder President, Sir, Dr. Chancellor, Sir, and our Vice Chancellor, Madam, I heartily welcome once again to the Amity University. Yesterday, we had a very fruitful discussions. And today morning also, we had a webinar where we discussed how India and Argentina can work together, not only in science, like typical institutional aspects, but also on cultural aspects, cultural exchange and science of the culture. So that's how we can collaborate together. And in this series, and we are happy that we have experts from diverse field. It's not only from the agriculture or science, but they are also from the medical science, public health, and sociology. So we can explore the possibility of collaboration not only in the field of agriculture and allied area, but in the health, public health, and medical science, and so on. With the same university, as Amity University is very diverse, we have 100 disciplines at our university. Similarly, University of Buenos Aires has also had multiple disciplines. So we are happy that a diverse, multidisciplinary delegation is here so that we can explore the possibility of opportunities. And I hope this lecture, which she is going to deliver, will give some food thoughts by which you can, which will stimulate you to do some kind of collaboration, not only for the research, innovation, but also for teaching or whatever way you can take it forward. So please be open during this and open your mind and listen with carefully so that you can get stimulated to get some new ideas or new thinking in your work, whatever you are doing. So thank you. With these words, I welcome both of you here for this video. And there are two lectures, one on NeoKit, another will be followed by another lecture. So I urge each one of you to have the patient listening. We will have the question at the end of both the lectures, not during the lecture. So that so please keep your questions ready so that whenever this discussion happens, you can ask a question. Thank you so much. So once again, I welcome you and thank you all for coming over here and giving there are multiple things are happening at Amity today. So despite that, you are here. So that shows your commitment. Thank you so much.
Thank you so much, ma'am, for your gracious and impactful words. Moving forward, it is my great privilege to introduce our distinguished speaker for today's first guest lecture, Professor Carolina. As Dr. B.C. Das has joined online, with my proud privilege, I now invite Dr. B.C. Das, sir, Dean Faculty of Health and Allied Sciences, Vice President, STEF, to please address the gathering. We would be truly honored to hear your valuable thoughts.
Bisida sir, are you there? Maybe later on, so you can.
Moving forward, it is my great privilege to introduce our distinguished speaker for today's first lecture session, Professor Carolina Carrillo, Principal Investigator at CONICET and faculty member at the UBA. Professor Carrillo is an internationally recognized molecular biologist with more than 25 years of research experience in Trypanosoma cruzi, the causative agent of Chagas disease. Her scientific career is dedicated to bringing the gap between laboratory research and real-world public health solutions. As co-founder of NeoKit SAS and director of research development and innovation at the ICT Milestone CONCET Kasara Foundation, she has led the development of simplified molecular diagnostic tools based on innovative isothermal amplification technology. One of her most remarkable achievements is the development and regulatory approval of NeoKit Chagas Neonatal, the first Argentine molecular diagnostic kit approved for early detection of congenital Chagas disease in newborns. Her technological platform also played a crucial role during the COVID-19 pandemic, enabling decentralized, accessible, and reliable molecular testing when it is most urgently needed. With this, her lecture is focused on NeoKit, turning a laboratory assay into a simplified molecular diagnostic kit for public health use, experiences in COVID-19 and Chagas disease. With this, I now invite Professor Carolina to deliver her lecture.
Good afternoon. Is it okay? Hola. Hola, hola. Okay. Well, thank you for welcoming us. And in advance, thank you for your patience because my English is so. So if I say something that is no understandable, please tell me, okay? Well, as Sumaya told us, I'm going to tell you a little short story about our work translating a lab experience into a product that is useful for health, for public health. Okay, as you know, infectious disease are major global have major global impact in public health and also in the productive sector. So one of the topics that are really important to manage the infectious disease are the methods of detection. So taking into account, we work in methods of detection for infection agents, but adapted to the characteristics of the individual, of the subject of study, the type of the sample, because it could be different if it's swab, saliva, or blood, and the infectious agent per se. But not only adapted to those things, also situated to the needs of the person. The needs of the subject will be studied, the possibility of the users, I mean the reality of the Is it okay? Yes. And not only adapted, but also situated to the real conditions in field. In particular, in countries like Argentina, that we are not a rich country, it is really important to be situated to their real life. So going to my topic, I work in Chagas disease. Chagas disease is caused by Trypanosoma cruzi. Trypanosoma cruzi, I think that it's better. Okay. Trypanosoma cruzi is an eukaryotic flagellated single-cell microorganism. Is far cousin of the malaria parasite. I know that you know just a little about Chagas. Chagas disease is more than a disease. We'd like to tell it a Chagas problem because it has a lot of condiments, not only health issues, but also social, economical, and cultural issues related. And these parasites affect mammalians, and it can cause illness only in 30% of infected people. The other people will be just healthy porter. Well, the transmission of this parasite could be by different ways. In natural ways or anthropogenic ways, but we are going to center in vectorial ways, that is by an insect we call kissing bug or vinchuca, that it transmits the parasite in its faces, not by the bit as the mosquito, instead by the faces. And it could be also the transmission by oral, taking infected food or drinks, and by vertical way during pregnancy or at the moment of the childbirth. And it could be caused also by blood transfusion and organ transplantation, but it can be prevented because we can test those organs or that blood before, and by lab accidents, that if we work correctly, we are not going to have that problem. So the most important ways around the world are the vectorial way and the vertical way. And going to the epidemiology, originally, Chagas disease was rural and Latin American because that kissing bugs were living in that geography. But taking into account that now we migrate and there are a lot of phenomena of spreading, so now is urban and global also. So it's supposed that almost 6 million people around the world, and you can see that in India we don't have, we have paint in pink the countries that have detected Chagas disease, and you can see that not only America, but also Europe, Australia, well, Japan, and we don't have any pink dot here in Bharat, but I think that probably here are some people with Chagas disease because we have migration, so it's possible. So I don't want to do some preoccupation. Instead, I'd like to think that Chagas disease is a neglected disease that should be taken into account at any point of the world. Respect to vertical transmission, it is one of the major causes, the new cases around the world. And in Argentina, that it's supposed that almost 4% of the Argentinian population is infected. We estimate that we have 20,000 of pregnancies carrying the infection per year. So it is estimated also that we have almost 1,000 of born infected babies per year. Well, taking into account this xenography, we can say that Chagas disease in babies by vertical transmission is a special charter because it's not an acute case, that it means that somebody that has born previously obtained the infection, at that moment, or no chronic infection, somebody that has the infection for a long time. So it is a special chapter. We are going to see why in different points. And most of neonates with vertical infection are asymptomatic. So it is really important that if we detect and treat early at those babies, they are going to be healthy people. So it's really important, the early treatment. So if it's important, the early treatment, it's really important, the early diagnosis. So early diagnosis is so essential for early and effective treatment. And so important is that in Argentina, we have two laws about the vertical transmission of Chagas. One is the newborn screening program that it says that every child born in Argentina must be tested for some metabolic infections, syphilis, and Chagas disease. And the other law that is so important for us is the law that it's called Prevention and Attention of Chagas disease, that says that every mother that is pregnant should be tested for Chagas disease, and that every baby born from an infected mother has to be monitored, and the notification is mandatory. So it's supposed that we detect all the cases with two laws acting. However, only 20% or less cases of Chagas in newborn are detected, are found per year. So you can imagine that there are a big problem, and the question is why? Well, we have some explanations. Some are biological and some are social, cultural, and economical questions. But taking into account this, available diagnoses have some difficulties because of the characteristics of the population of the babies, that they have variable parasitemia, and they have in blood the mother antibodies. So if I try to make a serological test, I'm going to find antibodies against Trypanosoma cruzi. But it doesn't mean that the baby has received also the parasite. And as I tell you, there are also barriers from the ground reality. Some barriers of the system of health to detect or not detect the parasite. Is it okay? You are understanding my English? Yes. Okay. So taking into account, our objective was to develop simplified tools for early detection of vertical Chagas in newborns. But translating this title, our objective was design a test sensitive, specific, and robust, but also simple and affordable, that could be used really in any place of our country. I mean, as I told you, situated in real life. So we based our project in the molecular reaction called LAMP, that it was described for a Japanese group directed by Notomi. Well, when we started to work with this, we conformed a public-private partnership consortium with our group of work, our lab group, formed in the Institute Milstein of CONICET, of the National Council of Science and Technology, and two companies that were interested in working with us to obtain that product. And we received an important grant from the Ministry of Science and Technology that, well, in 2011 existed and now doesn't exist. Well, and we started to work with that idea, but we proposed it not only as a test for Chagas disease, instead as a platform to detect different pathogens. So the platform proposed detecting pathogens because we can detect the genome of that pathogen. It could be DNA or RNA with high sensitivity, specificity, and robustness as a PCR or qPCR. Everybody knows PCR or qPCR here? Yeah. Well, most of us have learned about PCR after the pandemic. Well, in this case, the reaction is interesting because when we have the pathogen, we have some point of its genome that is specific. So any individual of that species is going to have that section of the DNA or RNA, but other individuals of other species are not going to have this section. So we select that part of the genome and design some primers that can be bind to that specific section. These primers at LAMP reaction has a special design that can do a loop, then we are going to see it. And if the primers can bind to the structure, then the enzyme PST polymerase can enter to the double strand of DNA and copy that structure. It is important because that polymerase is also helicase. So we don't need to change the temperatures at PCR or RT-PCR if instead we can do it in an isothermal way. So we have to put just 65 degrees, that is the optimal 64 degrees, that the enzyme can make the copies without changing the temperature. So the reaction can occur with a simple device, so simple, not expensive, and not complicated as the PCR or RT-PCR machines. Well, the reaction is a little complicated. We are not going to enter in details, but the question is that we can have thousands and thousands of molecules, starting with just one or two molecules. So one molecule or two molecules are not visible, but as a grain of sand, but you can see the desert. So when you have the DNA or the RNA present, you are going to have a lot of amplification. So we are going to see the mass at the final reaction. Well, so we work into the reaction itself, trying to select the best target sequence of the genome and working in the primer design, but also in the mixed components of the reaction and, well, the composition, the time, and the temperature for the reaction. But to be simple, because it is a molecular reaction, so at this point can be simple or not, the simplification is also taking into account how to manage the sample, what kind of sample we are going to use, and how we can obtain a good template from that sample, and the other parts of the reaction, how to reading out the reaction. Well, working in that way, well, welcome. Okay. Well, working in that way, we work in the performance of the reaction. I mean that it has to have a good sensitivity, a good specificity, and be robust, but also in the usability. So it has to be so simple and affordable and without complex equipment or complex infrastructure or so specialized personnel. And well, we were working on that, and we obtained in 2017 The first molecular test approved for ANMAT, that is our agency, regulatory agency like FDA in United States. And well, at this point, I can point out that it was the first molecular kit made by an Argentinian people. Okay. But there are some, it is the light part, but there are a dark part. But that it was a real, so proud design. But when we went to produce in an industrial scale,
Amy sounds like amity. Kids energy. So it looks like this is an amity platform.
yes, well, maybe it's a sign.
Yeah.
When we translate to produce in great scale, we have a lot of problems and it was so expensive. So the companies told us, well, we cannot produce it. We have to think about other ways to produce it because it is unaffordable.
So did you make it affordable later on?
Affordable that can be paid.
Okay.
Yes, because it was so expensive, the concept of production. So well, we start to try to change some things to do possible to industriality. So at that moment, we find, well, we fail in some cases and we start to find the best ways to produce. And at that moment, pandemia appeared in our lives. So as we had a lot of experience about what waste should not be taken, we could think about adapt that platform and obtain the kit for COVID.
Oh, very good. In
Yes.
43 days.
Yes, and it was after 45 days that it was declared the pandemic in Argentina that we obtained the test approved by ANMAT. So it was also so good, but I like to tell that it was 45 days because it was the formal time, but it was 3,287 nights because I count since we started to work with the first kit that couldn't be prepared. Well, in that moment, we also could install the manufacturing line and do the, yes, that production line.
So the production line was by the institute or by the company?
Well, as it is a public-private consortium,
Public
it is
sector.
inside the institute,
Right.
but it's a
Controlled by the
company. Yes.
company. Okay.
And
It was a startup or a proper company?
sorry?
It was a startup or a company owned?
It's
It
a
was
startup.
a startup.
Yes. Yeah. So that kit was used along all the country. More than 5 million of reactions were used in any kind of labs because it is really simple. So people that never have made molecular biology could use that kit. So you can see in these pictures a lot of people. We have more people, but I can señalar pointed that, for example, it was done in a bus that was a laboratory traveling around the country. So it was really simple. And when after pandemia went out, we went to my first log, Chagas disease, and trying to detect and conclude the kit that could be produced to detect Trypanosoma cruzi in neonatal population. And well, we obtained a kit that is really simple, that you can use drops of liquid blood or some dry drops of blood in a car. but it's a minimal volume that you need. So it's so important for a neonatal population because they have little drops of blood to be taken. And it could be used also with DNA, with purified DNA. But if you use DNA, purified DNA, you have to have the opportunity to purify it. So you have more abilities and capacities in your lab, but in some labs they don't have. And we have shown that the kit also function with the kissing bag faces or even with the rectal ampulla. It means that in some cases with you find the kissing bag that is dry or dead, and you cannot obtain the sample of feces, you can put that part of the body of the pinchuca of the kissing bag, and the reaction is going to work. Then you put that sample into a tube and put 1 drop of a disease solution that is a development of us that permit to obtain a good template, a good molecular template after making a pre-treatment at 98 degrees for 8 minutes. So after that, you obtain a sample that was pre-treated and is useful as a template. So you put into a reaction tube that it's also included in the kit, and you put a drop of another solution that is an enzyme mix that had the other components. When you put that drop there, you have a solution that is color violet. Then you make an amplification at 64 degrees for 90 minutes. And after that, you make the reading out at naked eye. We know that naked eye sometimes is not so a little heater that can read at real time. But it is a little most expensive than a naked eye. And as during pandemic, all people use at naked eye, we are sure that it functions well. So if the reaction maintains the violet color, it means that nothing happened inside the tube, so the result is negative. But instead, if it changed to blue, it's because you have the amplification and you have a chemical change inside the tube, so you have this change of color. Was recently approved by ANMAT, and it is producible, industriable, so we are so happy. And using the same platform, that I think that maybe it could be interesting for us, because thinking about collaborations, not only Chagas and COVID, we have also developed the dengue and chikungunya kit and monkeypox kit.
I think dengue is very important for us.
I think so. Dengue is important around the world, and we could develop maybe other
And
pathogens.
dengue and chikungunya both are important for us.
Yes, I think so. So, and we have also some kits for health of animals, like tick fever for the cows, and HLV, that is an infection from citrix that can be done with pieces of leaf or with the little insect that transmit that infection. So we are working also in other developments with different stage of development. So we can think about different pathogens that you can be interested. So here is the part of the group, of the NeoKit group. So Daniela.
Nice applied work. Very good, excellent.
I'm sorry.
Though we will take up the question answer at the end, but just quick one. Can we use this approach for the plant detection of the diseases and in plants also.
Yes, yes, of
Mm.
course.
Mm.
Yes, yes.
Similar approach, if we can develop some kind of quick strip methods for plant viruses and plant microbes like fungi, bacteria for
Yes.
diagnostics.
Yes, yes. In fact, for example, for cows, we have worked.
Because expression takes time. By the time the expression takes place, the
Yes.
infection has happened, but the expression is not there.
Yes.
So plant looks healthy, but suddenly it dies. So can we do early detection in the plants?
Yes, yes,
Mm.
okay. Yes,
Okay.
we with citrus we have been working.
With citrus HLB,
Yes,
yeah.
and it was so
You mentioned
useful.
about the HLB, yeah.
Yes, and you can do it near the tree, so
Mm.
you don't have to go to the lab. So it's really easy to do.
Yes.
You only need that little heater. Well, the reactives are maintained only at 4 degrees. They don't have to be fridge, and you can manage them at room temperature. And you have only 2 droppers. You don't have to be pipetting a lot of things, so it's really simple.
Yeah, good. Thank you. Thank you. Please sit down. There are plenty of seats here. Students, stand from the back. Please settle down. Thank you. So how many of you have heard of this Chagas disease? Can you raise your hand? Thank you. Anyone of you? Chagas? No. Chikungunya? Raise your hand. Dengue? How? And then what was that? HLB in plants? How many are the agriculture students here? Have you heard of the HLB disease in citrus? Citrus disease? HLB? What is the full form of HLB? Citrus, you know. Okay. How many of you know citrus? Citrus? Raise your hand. Citrus. I think everybody knows. Even the non-agriculture people know the citrus. Do you know? What is citrus? Or lemon, oranges. What are they? They're all citrus fruit. So there's particular disease which is caused by the bacteria, which is called HLB. And it has now transmitted to India also. Earlier it was only in US, in Americas, in American continent. Now it has transmitted to India also. So now people are talking about the HLB disease in India. Okay? And they have developed a kit to detect the HLB disease in citrus. So similarly, whatever. Acha, can you name one disease in plants? One disease. Anthracnose, very good. And powdery mildew is one. एक ही बच्चा है, सब नहीं पढ़ते क्या? What about others? Citrus canker, very good. Anyone else? Okay. I'm just giving you this thing. Just read about the HLB and then you will come to know what is this disease about. So, madam, the neo kit, madam Carolina, Dr. Carolina, she has an expertise in converting the DNAs, in taking the DNAs from the pathogen and develop the diagnostic kit. If any one of you are interested in doing that kind of work, if any one of you, you can always contact her and then try to develop any internship or some project, anything which you think of, okay? Or maybe you want to learn something, you want to understand something. So that is opportunity which you can get, okay? Even for the public health. How many are from the public health here? Oh, no. Okay. They have gone. Okay. Thank you. And we'll continue with the second lecture. So listen to the lecture patiently and then we'll have dialogue with each other. Okay. Thank you.
Thank you, Professor Carolina, for that highly insightful and inspiring lecture on translating molecular science to a more accessible form. Your work on transforming laboratory innovations such as NeoKit into impactful molecular diagnostic tool has truly enriched our understanding of translational research and its role in addressing global health challenges. With this, it is now my privilege to introduce our third speaker, Professor Mariana San Martino, independent investigator at CONICET and president of the Civil Association Hablemos de Chagas. Her lecture focuses on what if we look at complex health problems through a kaleidoscope, the example of Chagas disease in Argentina and beyond. Professor San Martino is a distinguished biologist and multidisciplinary researcher whose work bridges biology, social sciences, communication, arts, and education to address neglected tropical disease from an integral perspective with over 25 years of experience. She examines Chagas disease not only as a biomedical issue, but as a complex socio-environmental and cultural challenge. She holds a PhD in educational sciences from the University of Geneva and has served as consultant to leading international organizations such as the World Health Organization and the Pan American Health Organization, contributing to global strategies in information, education, communication for Chagas disease control. Today, she invites us to adopt a kaleidoscopic lens, one that recognizes health as an intersection of science, society, culture, and policy. I now invite Professor Mariana San Martino to deliver her lecture.
No presentation for me. I have some videos, but yes. Yes, but before I plan to talk a little bit, no videos or videos. So, hello, thank you very much for being here and for hearing us with respect and for this invitation and all the things, the good things that are happening to us these days. Thank you very much. So as she said, it's difficult for me to explain what I do and who am I, because I took pieces from so many disciplines. I used to say that I was a biologist. Now I don't know what, because I went to social sciences, communication, education, epistemologies, South epistemologies, and a lot of other things. Just to try to better understand the complexity of Chagas disease, knowing that trying to understand this, it's a way to understand health, to understand other health issues and other complex issues. So this lecture, well, I work in an institute of physics. It's strange. I work in a group of the science didactics that works inside the physics institute. So that will be for another moment to make contacts with them. They will be very interested, but I am in the didactic section. And also I work with Carolina in this association. She said that is Hablemos de Chagas. It's let's talk about, no, we talk about Chagas. Because Chagas disease, as other neglected diseases, is a issue that is nobody knows, and less people, not too many people talk about it. And when they talk about it, they talk only about some pieces and not the whole problem. And there is my job. And first of all, as a friend said, I have to say that I speak English very well, but the problem is that I only know 50 words. So be patient with me. Yes. So I will not use a PowerPoint presentation. I want to make today like a dialogue between some ideas and some videos I brought to show you different materials. And that's, I like to do that because I think also that it's a way that it is not only me talking to you. Through the videos and behind the videos, there are a lot of people, a lot of knowledges, a lot of languages that will be speaking today with me to you. So it's a kind of collective presentation.
in one person, and a lot of some videos. And also I want to say that these videos is a selection because we made a lot of materials that are at your disposal in internet, YouTube, only in Spanish, today with subtitles. I make a choose, but they are the result of complex and rigorous processes of research and dialogue of different knowledges. They are not the product of industry, or there is a lot of work, a research work, sometimes not visible, because when we think about research, it's easy to think about what Carolina does, but not easy to think that behind an educational material, there is a lot of research that has to be done with the same rigorous methodology of what you do in a laboratory. And I am saying that, and I would like, and I ask you to be attentive to 2 levels at the same time, if you can. The content, the videos are about Chagas, the content, but also about the characteristic of the material, because they have been developed for different audiences. They have different stories that I will not tell because I will be here on 2-3 days more, and but you can ask later. To think about, not only to think about Chagas when I talk about Chagas, but also to think about the issues that you like or that you are working on or you will like to work with. So let's see the first of them. Yes, the subtitles are so down. I think maybe you will need to stand up or try to understand the Spanish or. Sorry. No. The last one. Yeah.
A partir de este momento, es necesario que respires profundo, que te despegues de la idea y los límites de tu cuerpo material. Olvídate de las dimensiones conocidas. Te invitamos a entrar a un mundo microscópico y sumarte a un viaje muy particular, el viaje del tripanosoma. El Trypanosoma cruzi es el parásito que causa la enfermedad de Chagas. Es tan pequeño que solo puede verse al microscopio. Es un protozoo, un organismo de una célula que tiene además un flagelo. No es virus ni bacteria. Hay personas infectadas con el Trypanosoma en todo el planeta. Durante su vida, el Trypanosoma cruzi cambia de formas a mastigote, tripomastigote, epimastigote, para adaptarse a los ambientes por los que transita. Por su condición de parásito, vive dentro de otros organismos, entre ellos unos insectos conocidos como vinchucas y mamíferos, incluyendo a los seres humanos. Trypanosoma cruzi no vive en aves, anfibios o reptiles. Trypanosoma cruzi puede transmitirse por la vinchuca, transfusiones de sangre, embarazo o parto, por bebidas o alimentos contaminados, y raramente en el laboratorio o por uso compartido de jeringas. Trypanosoma cruzi no se transmite por la lactancia, ni por saliva o contacto físico como dar la mano o abrazarse, ni por relaciones sexuales. Volvamos a la vía vectorial. Las vinchucas se alimentan exclusivamente de sangre y por eso son todas potenciales transmisoras del Trypanosoma cruzi. Nacen de huevos, pasan por 5 estadios juveniles conocidos como ninfas y luego se convierten en vinchucas adultas. Cuando una vinchuca pica a una persona o mamífero infectado, se alimenta de sangre con parásitos. La próxima vez que se alimente, podrá transmitir el Trypanosoma cruzi, porque la vinchuca cuando pica, además defeca y en los excrementos habrá parásitos. Trypanosoma cruzi podrá entrar en el organismo a través de la misma picadura, al rascarse o por los ojos, boca, nariz o lastimaduras. Ahora sí, el viaje microscópico va a comenzar. Vamos al instante en que la vinchuca pica a una persona o animal infectado y el parásito ingresa a su interior. Los tripomastigotes ingresan a la vinchuca por el pico rostro y comienza el viaje. El flagelo, como un látigo ondulante, propulsa el movimiento. Trypanosoma cruzi avanza por el esófago. Y llega al estómago, donde diversas señales le irán indicando que debe transformarse. Se convierte en epimastigote y se prepara para encarar el largo y sinuoso recorrido del intestino. Los epimastigotes conservan el flagelo para moverse, pero además tienen la capacidad de multiplicarse. Por esto, durante el trayecto, son cada vez más, y al recto llegan miles de epimastigotes. En este nuevo ambiente, hay señales que indican que se acerca el momento de hacer un transbordo. Los epimastigotes se preparan, volviendo a la forma de tripomastigotes, que serán expulsados cuando la vinchuca defeque. Iniciando así una nueva etapa del viaje dentro de un mamífero. Los tripanosomas que estaban en la materia fecal de la vinchuca ingresan al torrente sanguíneo del mamífero. Dentro del vaso sanguíneo, el tripomastigote avanza, siempre con el flagelo por delante, junto con el flujo de sangre, compuesta por plasma, glóbulos rojos, glóbulos blancos y plaquetas. En algún momento del trayecto, el tripanosoma ingresa a una célula que puede ser del corazón, el sistema digestivo o el sistema nervioso. El tripanosoma debe adaptarse al nuevo ambiente intracelular. No necesita desplazarse y es el momento de multiplicarse. Pierde su flagelo y se transforma en amastigote. Los amastigotes se multiplican varias veces, llenando el espacio celular. Entonces, los amastigotes reciben señales para retomar el viaje. Se transforman nuevamente en tripomastigotes. Rompen la célula y vuelven al torrente sanguíneo. Cuando el ciclo de entrar a una célula, multiplicarse, romperla y volver a salir se repite. El Trypanosoma cruzi podrá causar daños de diversa complejidad. Esto ocurre en el 30% de las personas infectadas. Tu experiencia llegó a su fin. Pero los tripanosomas viajan y viajan entre vinchucas, personas y otros mamíferos, una y otra vez, y sus viajes son piezas que se suman a una trama más compleja, caleidoscópica, donde otros elementos entran en juego: derechos humanos, políticas públicas, aspectos socioculturales y mucho más, y se conjugan en torno a lo que conocemos como la problemática de Chagas.
well, I hope you understand. You can read would you read the subtitles? Yes? Oh, okay. And we have also this one in a virtual, with a shorter version for virtual reality lenses, and it's cool. And okay, well, I wanted to bring this just to talk a little more about this agent that Carolina works with and is in love with, but causes this complex health problem. And as I don't know if you see and you understood, I said that our association is called, in a short way, we talk about Chagas, but the real name, it's a big name and it's a question. What do we talk about when we talk about Chagas? It's strange as a name of a group, but we think that it's worth it to keep it because when we make science, sometimes we think that we know everything about the subject we work with, and it's not true. I'm sorry. There will be always other things to know. There will be always other sciences you need to better understand. There will be always the work and the knowledge of people outside the academy that is as much important as what we learn and do in our laboratory. And that's why we developed this, we call them a model to understand the complexity of Chagas and other health issues, but I will talk about Chagas. We started to think 15 years ago. This year we will be having our 15th party, like young ladies. I don't know if you do this in India, but in Argentina, yes. And 15 years ago, we will starting to ask between us, well, what do we talk about when we talk about Chagas disease? We know we will never arrive, be able to arrive at the answer. We will be answering and making one another time this question and always learning something and adding something to this way of thinking the issue. But we say, okay, Chagas is a complex problem, but if we say only that, it's maybe an empty words. We try to put this big thing that what is a complex problem into a metaphor that allow us to better understand it. And we arrive to the metaphor of a puzzle. Do you know puzzle? Puzzle, yes. If you are making a puzzle and one piece or two pieces are missing, you will never arrive to the figure, to the image you want to see or you want to finish. And we think that in health issues, sometimes there are a lot of missing pieces. There are a lot of pieces that we are only watching at. For example, the biomedical pieces, that it's okay, we need to know each time more about the biomedical aspects, but we need to search and bring to the puzzle also the other pieces that sometimes are underestimated. And that's why our puzzle has 4 pieces, the biomedical one, the epidemiological one that allow us to see the problem at big scale. Where are the people? Where are the insects? How much? Which are the incidents and this kind of index? And we have 2 more pieces, but the socio-cultural piece almost always underestimated and so needed as biomedical piece. That is the socio-cultural piece. This the one where we try to understand, for example, social representations, what people think about the issue, what people know about the issue, what are the costumbres? What are the habits of not only of people that lives with these problems, but also the habit, for example, of scientists, the habits of the teachers. It's so important to know that because sometimes they are obstacles that go beyond the biomedical knowledge. And the 4th piece we want to understand is the political piece. Not only thinking about the men and women that rules our lives and make the laws, but only the political part that touch us. The political understanding, not politics from ideology or partido político or parties, but also this political issue that keep us walking, that make us take our decisions. I will be a teacher because I want to change, blah. I will be a researcher because I want to solve or try to solve a problem. This way of understanding politics. It's okay, yes? You're following me? Yes? So before I follow, I will not, I have things to show. But I will not send it through the audience now because nobody will listen to me. So I will leave it there and we can show it later. But if you want, we brought with Carolina, it's a piece, these are the insects, the kissing bugs. Real, but in resina, in like a plastic, I don't know the word. And if you want to see. But yes. But they won't listen to me anymore if they have the kissing bags in their hands. They will add a photo to the kissing bag and and I have another thing to show that has to do with the title of my lecture, of my strange lecture. Because this puzzle issue, it's okay, but the puzzles, you have only one way to make a puzzle. And reality is more dynamic and the pieces interact. They are not only like a puzzle, like static thing, the pieces interact. The socio-cultural, political, biomedical aspects of any complex problem are in permanent interaction. That's when we add the metaphor of the kaleidoscope. Do you know what a kaleidoscope is? Yes?
Kaleidoscope?
Raise your hand. Who knows what is a kaleidoscope? 1, 2. My friends, 1, 2. Only 2.
No,
3.
there's one at the back also. Kaleidoscope?
Now you will know. This is a kaleidoscope. It's like an optical object. Beautiful. I will also give it to you so you can see through. You need to watch through the little hole. It's better if you direct it to the light and then go round. Yes, yes. So listen to me. It's important because it's complicated in Spanish. Imagine in English. I will do my best. So the metaphor. This issue, this thing has mirrors and has a lot of colorful beads inside. You will see beautiful shapes when you put your eye and you look like this. You will see beautiful shapes. And these beautiful shapes are made by what each of these colorful beads give to the system. Okay? So maybe you like more blue. So you like more biomedical sciences, for example. But you need to know that this color you like more, it's in a complex net. Yes. In a complex system that needs what has to bring the other bets. The other bets are the other disciplines, are the knowledge beyond academy, or the knowledge of people, everyday people, are what arts have to give us, that it's a lot. For example, we work a lot with arts. And also, the kaleidoscope tell us that there is sorry. There is there is dynamic. Nothing is static. And even I see this, I see a beautiful flower with, and I get hurt, there is movement. side. We will not see the same because we are different people, different observers, different ages, nationalities, blah. And that's another thing important to understand in these things. There is different, we have to take in account the different points of view. Okay? And well, go. Did you understand or you are saying that? Yes, 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 thank you. So you have the kissing bags, you have the kaleidoscope, and yes. And you please keep your attention in my person. Yes, 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 yes. Yes, yes. So you can, I can keep your attention. I will show you another video because we think, as I say, we think arts are very important. We work a lot with arts in each thing we do almost. And when I was talking about the dimensions, the pieces, biomedical, epidemiological, sociocultural, and political, we think that education and communication are essential pieces of the political dimension, of the political piece, because are the scenarios where things can move, when reality can be different, when we can imagine a different reality. So we think it's a model. We cannot, it's not necessary to agree with us. It's our proposal. We think that education and communication are essential to this piece. And that's why we work hard in these two scenarios. And that's why I know what I will show you something that you will caught your attention, because I know you love Messi. And now you are asking yourself, what does Messi have to do with all of this? This lady is a little crazy, yes. I will show you. So, okay. Sure. No.
Este es mi marido. Este es mi marido, Miguel Baltasar, un hombre valiente que siempre ha luchado por sus sueños. Uno de ellos era disfrutar de una gran familia y lo ha cumplido con creces. Un día me miró y me dijo:
¿Sabe cuál es mi mayor sueño? Conocer a Leo Messi.
Como muchos, Miguel nació y creció en el campo. Una chinche le trajo malos momentos y ahora es un futbolista quien le trae los buenos.
Cuando Messi era chico decían que era un líder en silencio. Pero nadie llega a ser el mejor jugador del mundo sin la valentía de ganarle a ese silencio.
Mi marido también ganó a su silencio. Cuando era joven, su inconformismo le hizo ir al médico. Siguió los tratamientos, se puso marcapasos y gracias a eso, mira dónde estamos hoy, cumpliendo el sueño de un niño de 85 años.
Nadie cumple un sueño si se lo guarda para él. Ahora es el momento de ganarle al miedo.
De ganarle al silencio.
Ahora es el momento de ganar a la enfermedad del Chagas.
Today, Chagas disease still affects about 10 million people around the world, mainly in the Americas, but also in other continents. Together, they could fill over 100 stadiums like the Camp Nou in Barcelona. In most cases, it is a silent illness, but it can cause diseases of the heart and digestive system. It is a neglected disease. It is therefore essential to get access to proper diagnosis and treatment.
So this spot, it's not new, and it's a result of research. It's not a spot made by an agency like from the air. It's the result of research we made with colleagues around the world, trying to understand what physicians, nurses living outside the Latin America, but in countries where Chagas disease is recognized as a problem, what they think about, what are the histories they have to tell. And that's how we met Mr. Miguel and his dream. And we were able to make, not me, but someone in the team were able to make his dream true. And well, I am almost finished. And I will show you the last, well, I was saying that we work a lot with arts and artists. I also bring to show you what some other materials. And you can see we each year we invite a plastic artist to make the draws of postcards that we give in our activities. So we do that since our beginning. So this year we will be having our 15th different set of postcards, and it's maybe as you don't know Chagas disease, there are a lot of things that you are missing. But if you understand, if you think about other health issues, a lot of times health issues are materials are too dark, or called to they make the point into death or into bad things. Okay, and disease is not a good thing, but there are a lot of, if you put color to your messages and diversity of aesthetic, and you invite to the table to the artist, your message will be much more listen. Thank you. That's why we think we need to work together. Well, also another material I can see. And talking about art, I will show you our latest material that will also show you music for our country, typical music, not tango, but folk music from other provinces, not Buenos Aires. And it was also raised. It has also a long story to tell. You will see the result, but I want to be insistent with that. Material doesn't appear like this. And okay, let's see the last one, and I almost finished. My friend.
Se va la primera.
Estaba observando el rancho de mi abuela la pituca. Debajo de la cortina se asomaba una vinchuca.
¿Dónde? Ahí está. ¿No la ves? Ahí.
Le pegué un grito a la abuela. Dejaste con la peluca, que este bicho sanguijuela lo voy a mirar con lupa.
Mirá, tiene 6 patas y rayitas alrededor de la panza.
Es un bicho peligroso, chupa sangre de animales, pica y anda por las noches, mejor que lejos lo mandes. Chipochicha, chirimacha, chincha, chinchera, vinchuca, que no se te suba upa, que no se te suba upa.
Se va la segundita nomás.
Me fui al doctor y le dije escuela pa' verla y ella muy pituca fue.
Abu, qué linda que estás. Te acompañamos.
Le hicieron unos estudios y no le encontraron nada. Mandaron blanquear el rancho con las grietas bien tapadas.
Esa vinchuca no vuelve más. Nunca más.
A mí también me estudiaron, me ha picado en noche oscura, pero como yo soy niña, me dicen que tiene cura. Chipo, chicha, chirimacha, chincha, chinchera, vinchuca, que no se te suba upa, que no se te suba upa.
Pensar que esta canción la hice hace 10 años con mi amigo Mateo cuando hacíamos el programa de televisión Chau Chagas. Hoy sigue siendo igual de importante hablar del tema, acá y en todas partes, no solo donde hay vinchucas. Porque hay vías de transmisión que no tienen nada que ver con las vinchucas. Y porque lo más importante es lograr que en cualquier lugar del mundo las personas que tienen Chagas lo sepan, reciban los tratamientos que necesitan y no sean discriminadas. Si quieren saber más, pueden buscar en YouTube los programas que hicimos con Mateo. Sigamos hablando de Chagas. Y no se olviden, el 14 de abril es el Día Mundial de la Enfermedad de Chagas. Nos vemos en el próximo programa.
Okay, I will leave it here. I will only say and finish that the kaleidoscope that is going around the room will stay at Amity.
Okay, thank you.
So maybe the kaleidoscope view help you to think another problems, another issues you work with. It's a good instrument, as good as the microscope. I recommend using for the research also a kaleidoscope.
Well, thank you so much. Thank you. So big clap for. Actually, she has highlighted how you can, about the social messaging, okay? Any problem, how to communicate to the society. So these are the ways by which you can communicate to the societies, like the movies or cartoons or the cards. She has shown so many ways by which you can communicate to the society the problems, the real problems. So for agriculture people, the farmers is one and consumer, okay? Suppose you want to take your research, suppose you want to take your message, whatever the message you have, to the society. So these are the ways of communication because these are the popular ways. This way, the message is communicated very easily. Otherwise, just writing if you give them the piece of the paper, it's not going to help. Is it? Even for you, I think even for the general public, for the students also, if you want to tell them, okay, you read this paper, how boring is it? Is it boring or it's interesting to read the paper? It's boring, no? But if I want tell you, okay, go and see the movie on the YouTube, is it interesting or boring? Interesting. How many of you love to see the movie on YouTube? Raise your hand. Netflix, okay. And how many would like to read the paper and think more? Nobody? You only. So this is how the message is all about. You want to give any message. It could be the education also, isn't it? Any education, any messaging to the students or to the society or to anybody. I think these are the tools which she highlighted, that how these tools are very impactful. And these tools cannot be like developed by one person. Okay, that's why the multidisciplinary is very important. These tools needs an artist, these tools need and people from ESCO. Isn't it? You know ESCO? How many of you know ESCO? ESCO, our communication, Amity School of Communication. Okay, so these so these are the people. So you need a team who can do the videographing. You need a people who can do the artists who can draw it. You may be some of you may be artist also. Some of you may be a video tuber, grapher, but you need a talented people to develop these tools. So if so you want to communicate like soil health, okay? You want to create an awareness among the farmers about the soil health. But farmer doesn't know about the soil health. Do they? If they know about the soil health, they will not do the mistakes. Will they? No, they will never make the mistakes. So you as a student, I give you an assignment, okay? You educate our farmers that they should not apply the fertilizer as per the recommended dose. What I'm going to tell you? Do not apply the fertilizer as per recommended dose. What does it mean? What does it mean? It means first they should test the soil. First, they should test the soil, how much is available. Recommended dose tells that the plant needs this much of fertilizer, but it doesn't calculate how much is already available in the soil. Does it? Recommended dose never tells you that what is already available in the soil. Okay, if recommended dose is 100 kg and 25 kg is already available in the soil, will you recommend 100 kg still? How much you will recommend? How much? 75? 75. So this is what your role is as a researcher, as an educator, or as a person who has the knowledge. So anybody can have the knowledge. It's not necessarily only the degree gives you the knowledge. Anybody can have the knowledge. If you want to transmit that knowledge to the common people, you need to have some tools. But if you want to tell the farmer, okay, आपको 100 kg नहीं 100 के इतना करना है, will he able to understand it? No. You have to make it understandable. To make it understandable, you have to take the help of these tools. This is what the message is of this particular presentation. The tools, communication tools are very important. Okay? Communication tools are very important even for your own disease. Like for example, we want to create an awareness about any disease, social disease or any disease. We use these tools. So this is what the message is all about. And they have successfully experimented that these tools really work and the awareness can be created. So those who are from the public health can think about the diseases, the public health, any health issues which are not, people don't know about them. Like during the COVID time, you remember how many times we used all tools to create awareness about the COVID. Wash. I think there was a song which came the wash 40 minutes or 30 seconds or something like that. Huh? The song was created. There was a tool which was created in your mobile that do this, don't do this. That's how the all awareness was created. Otherwise, how will you come to know about it? Similarly, for those diseases, for those issues which are still neglected, you can use these tools to communicate properly. So this was the message for this particular message. Thank you so much. And probably there's more scope for communication, more scope for interaction, and more scope to learn from each other's experiences. Thank you so much.
Thank you so much, Professor Mariana, for your enlightening talk and informative videos. Your perspective on importance of understanding and talking about Chagas disease enlightened us to rethink the health challenges we face as a society. The theory and your perspective on kaleidoscope you presented that represents the diversity in society, communication, education, and science provide us a new way of thinking and broaden our understanding of public opinion regarding health challenges. With this, I now invite our Director General, Dr. Nutan Kaushik ma'am, and Joint Head, Amity Institute of Pharma, Dr. Tanveer Naved, sir, to please felicitate our speakers. Professor Carolina Carrillo.
Oh, thank you. Yes.
Please, ma'am, please felicitate Professor Mariana San Martin. With this, we finally come to the conclusion of an enriching two-day international lecture series hosted by Amity University, Uttar Pradesh.
I think all the faculty can come forward for the group photo. Please. And those who have worked very hard, those also.
Nothing. Yes. Public health is good. Delhi side, all public health. Fatafat. They want some photograph. Okay. Ah, yes. Yeah. That's gone. Thank you. Thank you, everybody.